is practically worldwide. I mean, he's got programs in Madagascar, in his native Florida. Those of you who got the native orchid stamps that were offered by the post office, there's a very rare Platanthera leucophea that's critically endangered. They work on that, and he also does work on Cuba with ghost orchids. So the Orchid Recovery Program is something really special and really unique, and I'm just so pleased to be at all involved and to be able to be even a little helpful to a program like that. I think it speaks towards a better world and a better planet. So bravo to you, Larry, and to all your students.
You're going to make me cry there, Tom. I might have to have a hug here. So
soon we will all be able to hug each other.
Yeah, can you imagine if over Zoom suddenly we can like feel each other?
For real, yeah, for real.
Could be weird.
And so I'm just going to say a couple housekeeping things before Molly starts, in case you don't know. Most of you know probably, but if you tap your screen, somewhere on your screen, you can go from the grid view if you just want to see the face of the person who's speaking and you don't want to see all the little thumbnail things. You can go back and forth. Also, please keep yourself muted during the presentation. It helps for all of us to have better audio quality, so there aren't these funny noises in the background, or kids crying, or dogs barking, or cats meowing, or whatever. And rain, it's raining too, which makes a noise on the tin roofs. So, and also somewhere on your screen, you'll be able to see how many participants and who's here. And I can see that most everybody is muted. Some people have decided not to turn on their cameras, which is a shame, but that's up to them. So, and then there are little places, if you go into where it says other, it will show you that you can clap. There's little emojis for clap, a thumbs up, a heart, laughter, and other things. So, and it will show up briefly in your square. If you want to clap for Molly, I just usually clap like this.
Yeah. There's a tab for reactions at the very bottom of your, all the way over on the right. And so you have all those choices.
Oh, it just depends on what device you're using. For on an iPad, for me, it's up in the upper right-hand corner. But it's going to be different for everybody where all those little things are on your screen. Well, welcome, Molly. We're interested to hear what you have to tell us, and I'm gonna pass the baton over to you, and you can speak as long as you want.
All right. Well, hello, everyone. I'm just gonna start sharing my screen, and then I'll get started.
Okay.
Can everyone see the screen okay?
Mm-hmm.
All right, sounds good. Hello, everyone, and thank you for coming to hear about the updates concerning the Orchid Recovery Program at Illinois College's progress with the federally endangered Peristylus holochila, the Hawaiian bog orchid. I'm Molly Gearin, and I've conducted the counts of the plates with the help of others in Dr. Zettler's lab. This is my first year working in Dr. Zettler's lab, but I've been able to gain a lot of experience and have worked primarily with Peristylus holochila and its asymbiotic germination. Before we get started, I have two things to mention. First, I would like to recognize Dr. Capo, who is the director of the Illinois College Honors Program. She's listening in to this talk, which is the completion of my honors capstone presentation requirement, sharing my research. Secondly, I'll admit that I do have trouble with the pronunciation of some of the Hawaiian words and locations, so feel free to chime in and correct me or help me out. So currently I am located at the Illinois College campus where I care for Peristylus holochila 6,600 kilometers away from its native habitat. Illinois College has been working with this orchid since 2002. Illinois College is located in Jacksonville, Illinois. From this map, you can see that Jacksonville is located in central Illinois, away from larger, more well-known cities, including Chicago and St. Louis, Missouri, where I'm from. The city limits of Jacksonville border cornfields, and yet at Illinois College, some of the most rare orchids are cared for in Dr. Zettler's lab. While most people are unfamiliar with Illinois College or assume you're referencing a generalized college in Illinois, Illinois College is the oldest degree-granting college in the state, founded in 1829. It's a Phi Beta Kappa institution, and despite being relatively geographically isolated, is a world-renowned institution for many of its professors and students' accomplishments, including Dr. Zettler. Dr. Zettler's lab is part of the National American Orchid Conservation Center, which is a network of institutions that work to grow and reintroduce orchids in conjunction with the Smithsonian Environmental Research Center. The NAOC focuses on Native American species in the greatest danger of extinction and focuses on reintroduction of seedlings, emphasizing germination using mycorrhizal fungi. Now this map shows all of the institutions which are part of NAOC, or the National American Orchid Conservation Center. These institutions include Illinois College, the Chicago Botanic Garden, the United States Botanic Garden, and the University of Alberta. The Orchid Recovery Program is the title of the lab at Illinois College, which works to reintroduce threatened orchids. At Illinois College, as part of NAOC, we also focus primarily on maintaining the most natural germination, which is by using its native fungal symbiont whenever possible. I'll now give a basic overview of orchid biology, which should be a good review for experts and enough of a basis for newcomers to grasp the situation surrounding Peristylus holochila. From this picture, you can see that orchid seeds are extremely small and are almost dust-like. The image on the left shows a range of orchid seeds. Some have increased surface area, which acts as a sail, suspending them in air as they travel. The image on the left shows the clear seed coat surrounding the embryo. Healthy embryos are nearly colorless. Now, orchid seeds contain no endosperm. This means that they have no food source and need something to get carbon from in order to grow and survive. This is where a fungus comes in. The fungus supplies the carbon necessary for the orchid to grow and survive. In nature, the hundreds or thousands of orchid seeds are released in hopes of landing near a fungus which can host the seed, which is why the seed's air suspension is so important. This image shows the massive fungal hyphae, or the peloton, which has invaded the embryo. The mycorrhizal symbiosis of fungus in an orchid is not a mutual symbiosis, however. The orchid is actually the aggressor. In mycotrophy, the orchid seed actually feeds on the fungus. The fungus provides the food or the carbon for the orchid to grow. The fungus also provides water, which is necessary for seeds to prevent from becoming too dry and dying. However, fungi do not need orchids, but orchids need fungi. The orchid is the aggressor and it needs the fungus to grow. With a secured food source, orchid seeds begin to grow, becoming protocorms or leafless orchid seedlings. From the 2 millimeter reference, you can see that protocorms are still extremely small, but with the right fungal symbiont, seeds can develop leaves in a matter of months. This image depicts how protocorms develop a leaf shoot. The region at the base is the mycotrophic region, which continues to act as a food source for the orchid. But now the leaf has developed so the orchid can also receive carbon from photosynthesis. The orchid can now use 2 methods to take in food, producing its own and taking carbon from its mycorrhizal symbiont. The idea of finding the right mycorrhizal symbiont will be important as we now turn to Peristylus holochila. Peristylus holochila is known locally as Puahala akani, or by its common name of the Hawaiian bog orchid. Peristylus does not look like a signature orchid. This disguise adds to its personality as an orchid which is difficult to sow experimentally, but which has so far managed to fight extinction despite being on its brink for many years. As I mentioned earlier, Illinois College has been working with Peristylus holochila since 2002. Before this, the University of Hawaii was conducting experiments with germinating this orchid. Dr. Zettler is known for his specialization of in mycorrhizal fungi, and for this reason he was contacted by Steve Perlman to help in the germination of this orchid. However, this orchid is particularly resistant to symbiotic germination with its fungal symbiont. For this reason, my study concerns seeds sown without a fungal symbiont. Fully mature, the orchid is green with inconspicuous flowers which can number from 5 to 60 per plant. It has a pleasant light fragrance, despite being assumed as a self-pollinated orchid, and there are approximately 1,000 seeds per seed capsule. Peristylus holochila bears a close resemblance to Platanthera hyperborea, which is native to Iceland and Greenland, but has been recorded in Alaskan bogs of high elevation. The Pacific golden plover migrates between the Aleutian Islands, which is an island chain in Alaska, and the Hawaiian Islands. It is hypothesized that the Pacific golden plover may have carried the small orchid seeds on its feet from Alaska to Hawaii long ago. These two plants are considered genetically unique, and that if this hypothesis is correct, that Peristylus holochila speciated once it arrived in Hawaii. Nevertheless, Peristylus holochila is recognized as a distinct species from Platanthera hyperborea. Peristylus holochila has been listed as federally endangered since 1996. The current extant population is found only on 3 Hawaiian islands. There are 6 plants on Maui, 1 plant on Kauai, and a few dozen left on Molokai. The state of this species' survival rests on the seeds left in Dr. Zettler's lab. In its native habitat, Peristylus holochila is found at high elevations from 600 to 1,500 meters. The habitat is cool in temperature around 10 to 19 degrees Celsius. They're located in mountain bogs high in organic material and acidity as described by Dr. Zettler and Steve Perlman in 2011. The acidity of the bogs has a pH of 4.3. The exact locations of the remaining plants of Peristylus holochila are protected to prevent illegal harvesting. The Hawaiian Islands are subject to many of the same threats that we have on the mainland, including climate change, natural disasters, and invasive species. Many Hawaiian native species have already been forced into extinction, but the Hawaiian bog orchid has escaped this fate so far. On the mainland, we have heard about the invasive species which threaten native species, including the feral hog and axis deer. These animals further endangered the survivability of the species already pushed to the brink of extinction. These threats further emphasize how vulnerable the few remaining plants of Peristylus holochila are and how important the current attempts to cultivate the plants for reintroduction is. In the experiment that I study, there are 4 seed sources that I'm looking at. Each seed source has a unique number which is preceded by the letter S. This follows the organizational protocol of Dr. Zettler's lab to distinguish fungal sources from seed sources. The seeds from each of the 4 sources were sown on June 30th of 2020. 3 of the seed sources come from the sole plant on the island of Kauai. 1 of the seed sources originates from the island of Molokai. The seeds I study were sown asymbiotically because this orchid has a history of being difficult to propagate symbiotically from seed. Starting in 2002, Illinois College, along with the National Tropical Botanical Garden of Kauai, attempted to sow Peristylus holochila with fungi drawn from their roots. This first attempt yielded a new species of Tulasnella discovered by Dr. Zettler, but this fungus did not successfully grow with the seeds of Peristylus holochila. Peristylus holochila successfully grew with the fungus Tulasnella calospora. This fungus came from the orchid Spiranthes brevilabris, which is from Florida. The fungus was known to be successful with several other North American orchids, so that's why it was selected to try with Peristylus holochila. It was exciting that this was successful for Peristylus holochila because it finally grew symbiotically from seed. This fungus was native to Florida, so it could not be reintroduced to Hawaii. There were concerns that introducing a non-native fungus in an already vulnerable habitat would leave this to not be a realistic method to use. So after this, the lab pursued asymbiotic germination, as it seemed to be the best path forward for reintroduction, given an orchid couldn't be introduced with a non-native fungus. This led to 86 seedlings being brought back to Hawaii. Later attempts continued to pull for fungi that could be successful experimental symbionts for Peristylus holochila and provide a faster route for reintroduction. The new idea the lab tried was to use a naturalized orchid as a sort of environmental filter to draw the fungus out. This would leave the established Peristylus holochila untouched from isolating fungi from its roots. The naturalized orchid could be completely removed and could have the fungus lifted from it to test its viability with Peristylus holochila. In 2017, Hank Oppenheimer used Polystachya concreta as the naturalized orchid at Pu'ukuakui Preserve. This pulled another Tulasnella species, F432, which was used with symbiotic germination for Peristylus holochila. But again, it didn't work. So again in 2019, Hank Oppenheimer attempted to use the orchid Zeuxine stratomatica, a well-known naturalized or terrestrial orchid. This experiment was conducted at Maui Nui Botanical Garden, and it yielded F435, another Tulasnella species. This fungus also did not work in symbiotic germination with Peristylus holochila. So again, in 2019, Hank Oppenheimer used the orchid Spathoglottis plicata, which was naturally located near a Peristylus holochila individual at Waihi'i Valley. F436 was pulled from Spathoglottis plicata, which was another Tulasnella species. But again, it did not initiate symbiotic germination with Peristylus holochila. So germination without fungi was pursued once again. There was no viable fungus to use for symbiotic germination, so this type of germination was successful 10 years ago, so we tried to repeat it. This picture shows seedlings that were brought back to Hawaii 10 years ago. This method of germination was seen as the best path forward for Peristylus holochila. Asymbiotic germination uses the agar as a food source for the seeds to use for growth, bypassing the need for a fungus. This current experiment aims to reintroduce as many seedlings as possible. However, sowing the seeds without mycorrhizal fungi significantly lengthens the growth period. It takes an orchid nearly 3 years to reach the maturity level required for reintroduction when sown asymbiotically. In addition, upon deflasking, the survivability is significantly lower than those sown with mycorrhizal fungi. However, asymbiotic germination has been the most promising path toward reintroduction for Peristylus holochila when other options are not possible. I'll now detail the methods used for this experiment. The process for caring for Peristylus holochila seeds when they arrive at Illinois College is crucial to ensuring their possible reintroduction in a few years. The seeds are harvested just before dehiscence. Missing this key time can lead to premature seeds or increased contamination when capsules burst. When the seeds arrive in the lab, they are placed on dry rite, a desiccant, for a week. Following this drying period, the seeds will be carefully extracted from the capsules and placed in a glass vial where they can either be frozen or proceed with sterilization. The seeds that I study were cleaned and sown on June 30th, 2020 by Fagor MEI. The seeds were surface sterilized using a 5% bleach, 5% ethanol, and 90% sterile distilled water solution. The seeds are placed in the solution and shaken vigorously for 1-minute intervals. Then liquid is removed from the solution, leaving the seeds. Then seeds are rinsed twice in distilled water. This process is repeated several times until all visible contaminants have been removed from the solution. Then seeds are placed on the agar surface of a petri dish using a sterile wire loop. They are then wrapped with a layer of parafilm to contain the moisture while protecting against contaminants. Plates are kept at 16 to 19 degrees Celsius in complete darkness. They will remain in this state for around 450 days. They are only removed from these conditions when I conduct my counts or when they are checked for contamination. The last count I completed of my seeds was done on their 224th day of their 450-day time in complete darkness. Seeds will proceed to the next step once they are leaf-bearing. These seeds were sown asymbiotically on acidified P723 agar. This was found to be the best carbon-based media for Peristylus holochila's success. In previous comparative studies, P723 successfully grew seeds to stage 5. Prior to sowing the P723 agar, the pH was lowered to 5.2 because of Peristylus holochila's natural habitat being in acidic bogs. HCl is added to the agar to raise the acidity to mimic their native soils. The seeds I am studying come from 4 different seed sources. One of the seed sources, S209, comes from Molokai and was collected in 2012. The 3 other seed sources come from Kauai and were collected in 2013, 2018, and 2019. I began counting the seeds in August of 2020 when they were beginning to germinate. Since then, I've continued to monitor their growth. They are assessed using a 0 to 5 growth stage as described by Zettler, where stage 0 denotes no germination, stage 1 denotes rhizoids evident, stage 2 denotes rupture of testa by enlarged embryo, stage 3 denotes appearance of a shoot region, stage 4 denotes emergence of leaf from shoot, and stage 5 denotes leaf elongation. From the graph in front of you, the dates that I collected the data are on the x-axis with the bar per date representing each seed source's germinated seeds, updated last on February 8th, 2021. On the y-axis is the number of germinated seeds counted. Each bar is separated into each stage present. Seeds in stage 1 are indicated by the orange bar, seeds in stage 2 by the yellow bar, and seeds in stage 3 are indicated by the green bar, which again indicates that they have begun to develop a shoot region. None of the seeds have entered stages 4 or 5 yet, and that's yet. They're still growing. An overall trend you can see is that the overall number of germinated seeds has increased from August to February. In addition, you can see that seeds have not only germinated, but have continued to develop over time for most of the seed sources. Seed source S217 has the most germinated seeds of all sown seed sources in my experiment. This seed source has the highest bar for each of the dates. So far, it has been highly successful in continued growth and progression in germination and development. From the last count conducted, there were 19 stage 1, 61 stage 2, and 25 stage 3 from the S217 seed source. This table shows how many of each stage are present from each seed source. The top row of the table shows each of the stages present, and the first column shows each of the seed sources. These data were updated as of February 8th, 2021, which was their 224th day since being sown. So far, there are a total of 172 germinated seeds among the 4 seed sources in this experiment. This far outnumbers the plants in the native habitat. This is incredibly exciting. However, there are still many risks facing these seeds' survival that will ultimately determine how many seeds will be reintroduced once they reach full maturity. This table documents the percent viability and percent germination of each of the seed sources. Viability is the number of seeds containing embryos divided by the number of seeds with and without embryo. Each of the seed sources has high viability. The percent germinated is the number of seeds for each seed source which reached at least stage 1 divided by the total number of seeds. What is important to note is that the 36% seed germination of seed source S217. This is interesting to note is that the seed source S217 has the highest number of germinated seeds, including stage 3 development, and the greatest germination percentage. They were collected in 2013 from Kauai. What could be indicative of the seed source greater germination rate could be that the seeds were collected at the optimal moment. One of the most crucial factors for orchid seed germination surrounds the date of collection of seed being the most optimal time before dehiscence or the bursting of its capsule. Expanding further on the S217 seed source, these seeds were collected from the sole Peristylus holochila plant on the island of Kauai from the Alakai Bog in 2013. These seeds spent 6 years, 7 months, and 15 days in the freezer. This exceeds what was originally thought to be the past seed viability of 5 years in the freezer. Seeds which were collected after this date did not even achieve this high of a germination rate. As noted before, this seed source has the most germinated seeds of any of the seed sources in my experiment. The single plant on Kauai is decades old, and each year could be its last batch of seeds. This is why it's so important that these seeds are cared for and continue to develop so that they can be reintroduced. This slide shows the time spent in the freezer for each of the seed sources. S209 spent the longest time in the freezer, and from the one plate that I monitor, there are seeds that germinated, but they have not continued to grow so far. S217 spent the second longest time and has obviously been successful so far with germination and continued growth. S231 and S236 both spent much shorter times in the freezer and have germinated seeds which continue to grow at this point. Here are some images of germinated seeds from my experiment. The white color is indicative of a healthy germinated seed. On the left side, you can see a stage 3 seed. Rhizoids extend out in a point, which is not nearly visible with the exposure of the light, but there's a point at the top. On the right side, you can see that seeds have turned brown or black around the white seeds, which is indicative of death of the seed. But the middle seed has a clear shoot region pointing out as their stage 3. Now, once seeds develop a leaf, they will be moved from darkness to 12 hours of light and 12 hours of darkness, where they will remain in their original place for an additional 200 days. Following these 200 days, the leaf-bearing seeds will be moved to a new plate of acidified P723, where they will remain again in 12 hours of light, 12 hours of dark for 12 months. Once the basal leaf has greater than 2 centimeters in length, seedlings can be deflasked and placed in a new container with a 4:1 ratio of carnivorous plant mix, ground tree fern, and builder sand. Seedlings will remain in these conditions for 2 to 6 months in cool temperatures, exposed to natural illumination. Seedlings will later be introduced to higher elevation habitats from February to April. Justine Kennedy, a sophomore, and Tony Ruiz, a first-year student, have been conducting counts of a new batch of Peristylus holochila seeds, which were sown last November. They were cleaned and are kept in the same conditions as I described before from my experiment. These come from 3 different seed sources. Some of their seeds come from Maui, the third island where Peristylus holochila is found. Justine and Tony have just completed their initial counts of these plates, which have started to germinate at stages 1 and 2. Since I'm a senior, Justine and Tony will be taking on the seeds from my experiment when I graduate, and may be the ones who get to plant the seedlings in Hawaii when they reach full maturity. I'd like to thank many people who have made this research and presentation possible. Thank you especially to Dr. Zettler and to all of the contributors and donors listed. I'd like to thank the Hilo Orchid Society for their generous contribution. And I'd also like to thank Illinois College alum Doug Sutton, whose donation allowed us to purchase an emergency generator, which is incredibly important for the freezing of our seeds. I'd also like to thank Tom Marinda, who invited me to speak today to the Hilo Orchid Society. And I'm willing to take any questions. Me and Dr. Zettler will answer them for you.
Thank you so much. Yeah, very well done. So this is a good time if anybody has any questions for either Molly or Dr. Zettler. This is our question and answer time. Yeah,
I
Tom.
have a couple questions.
Okay.
So the plants are so rare in their natural habitats, but they do go to seed. They're self-pollinating plants, you're saying? They're not really dependent on a local pollinator. Why do you think the populations, first of all, are so small? And has anyone gone and done like a transect to see if natural seedlings are germinating? And if they are, great. If they're not, why? And of course, that's why you are trying to propagate the seeds, because the populations are so small. But I'm wondering why it doesn't naturally happen.
Hey, Molly, you
Want me to answer this?
give it your best.
I'll give it a shot. So I think that the surrounding situation for Peristylus holochila, there's a lot of pressures from the environment that are stressing the individuals. They obviously are in very low numbers, and I think that the pressure from different natural disasters and invasive species are threatening them further. They are self-pollinating because there's so few pollinators in their natural habitat. I personally have never been to Hawaii, and I don't know of any transects that have personally gone to look at the populations. So maybe Dr. Zettler can answer that part.
Tom, that's an excellent question. And I've been doing this for 30 years with these orchids, terrestrials and epiphytes. There's one pattern I'm beginning to see now. In fact, with Platanthra leucophaea here, I think that some of these orchids, and I think this one might be one of those, the seeds just aren't getting there. I think it's a system where we have to have the right fungus, obviously, to initiate germination, but the seeds just aren't getting to the right places, enough of them. And I say that because back in 2002, we put some seeds in packets on Molokai, and we got germination within a year. Baby holochila seeds were in the packets. So if you get the seeds out there, they should be there. Now, getting them to the leaf bearing stage is another matter. Maybe there's, maybe there's a drought year. I don't know. I also think that the habitats are very specific for this orchid there, where you're at it. It likes higher elevations. I think, where it's cooler. If it's really true that this orchid comes in from Alaska, well, the ancestors of it, that would make sense. So it's probably a collective of things, but I think that maybe the seeds just aren't getting there as one of the primary reasons.
I'll just, for the sake of our members, I just want to explain what Larry was talking about with the seed packets. There's a process that they use with native terrestrial orchids that they call fungal baiting. And basically they take the seeds that are produced by the plant. Oftentimes terrestrial orchid seeds won't germinate right next to the parent plants. And in fact, the mycorrhizal fungi that are associated with adult plants actually discourage other seeds from germinating around them, which is a defense for the parent plant. So they take these packets of seeds and put them some distance away from the parents, but in similar habitat. And when those seeds then germinate, they can test for the mycorrhizal fungus that actually germinates the seeds. So what Larry is describing is a process called fungal baiting, which is really a fascinating and ingenious idea that I think originated with Dr. Wiggum at the Smithsonian. So anyway, so the fact is, on Molokai, were you able to extract fungi from the ones on Molokai?
Yeah, so that was so baffling, and that's why I have so many gray hairs now. The protocorms gave us the fungus. It was hard to get. We think it's a new species because we did some DNA work on it. It turns pink in culture. My daughter's favorite color is pink, and I'm going to probably name it after her. But on the downside, it didn't do anything. It didn't germinate the seeds like we had hoped.
This is the burden of being the child of a botanist. You get fungus named after you.
That's right. You get yeah. Well, at least it's not a pathogen. So and so yeah. So anyway, I don't know why. I'm. What can I say? So we went the asymbiotic route, and we're just thrilled we are getting some seedlings again, and so we're back. So we hope to get these out there in another 2.5 years. I want to mention that 2 of the folks that are going to take care of these babies that Molly has now are on our view here. They're listening in. So Tony and Justine are both here, and they're going to carry on, and hopefully they will have a chance to take them back to Hawaii before they graduate. That's the plan.
Well, maybe when you come, you can come to the Big Island. This is a pretty big island. Maybe we can find some high elevation bogs where they might be on this island as well.
Never been to the Big Island. I'm eager to get there.
Oh gosh, you're missing out.
I'm pretty shocked that there are none of these orchids on this island, because we have such high elevations here, and we do have boggy areas as well.
It's probably here.
Yeah, I would think that it probably is, and we haven't found it yet. And Molly, I just wanted to say you were doing a pretty fantastic job there with those Hawaiian words, and it's just a, e, o, u for the, and if there's 2 vowels side by side, you pronounce them both. you know.
The glottal stop in between.
Yeah, yeah. So, and then I think Larry Kikas had his hand up a while ago. Did you still have a question, Larry?
Nope.
No. And then Kathy has her hand up.
You're still muted. Kathy.
You're still muted, Kathy. And then we have somebody else with their hand up as well when Kathy's done.
Okay, years ago, we're probably talking 20 plus years ago, 25 years ago, right when GPS units first came out, Hilo Orchid Society, I don't know how we got into doing it, but we had heard there were some of the native Hawaiian orchids over in Puna somewhere. And I'm not sure who made contact, but we made contact with a group of pig hunters. And they used my phone number. I was the contact person. So I was getting these strange calls, you know, from Thick Pigeon. Oh, you the lady stay looking for the plants. Anyway, we got a couple of seed pods and gave them to Carmela. Sheldon said they would germinate them. And they did get them to start growing in the flask. And we used to visit them a couple times a year. We used to go into the lab and they started to grow, but that was before the fungus was discovered. We didn't have the right growing conditions. And they were growing probably for a year or two, and then they just started looking worse and worse. And I think it was a total of 5 years that they finally ended up not living, but we had them in the lab in Hilo at Carmela's Orchids.
I wonder though, Kathy, did they say these were native Hawaiian orchids, or were they like one of the Spathoglottis or?
No, they were
orchids?
definitely, but I think it was, is there a Plantera?
Platanthera, which is the same.
Plantera?
Peristylus is the genus that they moved.
Okay,
Or native.
it
So
was one of those.
and it was here on the Big Island. It was
over in Puna. I know, I just remember because once the pig hunters marked them, 2 of our members who now couldn't go traipsing through the forest, but they went into the forest with a couple pig hunters and marked it on the GPS. They had a handheld unit. Our club bought a unit back, had to have been mid-90s. And like I said, they went in a couple times until the pod was ripe, and then they harvested it and brought it to Sheldon.
Well, I think it would be very worthwhile to get that that GPS data, if it's possible to get it from them at this point, because that could be a whole other population that that Illinois College might be interested in using.
That's true. I know one of them was Larry Ford, Brenda Ford's husband. He was one of the people that went into the forest. So I can ask if they have the records at all.
I wish you would. Thank you.
Yeah. Okay.
Thanks, Kathy.
Yeah.
Hang on, Phoenicia. There's somebody else that also has their hand up before you. Guy, I can't see your name. You have a red shirt on.
Oh, okay. I'm Ingo White from Windward Community College on Oahu. Considering that you were successful in germinating that seeds is so fantastic there, because I know it is very difficult to germinate the Hawaiian native orchids. Do you have any plan? What kind of effort would you like to do when you introduce the seedlings back to nature in Hawaii in order for them to be 100% successful growing in the bog? After all your work, you must have some kind of planning to protect them in such that they will continue on growing when you introduce them back to nature.
Yeah, one of the reasons why we like that we regrow native orchids, especially terrestrials, they're more difficult to establish than the epiphytes as a group. And so that's one of the reasons why we were asked to grow this orchid from Hawaii, because it's a terrestrial, because we use fungi. Fungi help to establish the connection, and the survivability goes up usually. So that was our ultimate goal, but we didn't have that luxury because the fungus just didn't want to work, and neither did the other fungi that we've been trying. So we were there in almost exactly to the day 10 years ago. And the night when the day after we finished putting all the orchids in the ground, we were hit by the tsunami, the Fukushima one. And that's a long story. But also some of these plants, a subset were taken over to Maui, to Olinda, and Hank Oppenheimer's care. And a few of them flowered in the pots. And so we are, as expected, without a fungus, you don't have very good survival. And I don't know the fate of those that were put on Kauai or Molokai. I don't have the numbers. I don't think they made it. But a few in the pots from Olinda on Maui did. They flowered. So that gives you some hope that, once you get to the flowering stage, that you have the possibility of having siring, spontaneous seedlings. I was also really, glad that they flowered, because we were very careful about putting numbers on the seeds we get and we put in storage, and it would be kind of terrible if they showed up and they were spiranthes or something, not the real thing, but they were the real thing, and we were sweating bullets. No, we were good. We were very careful.
Ira, you had a question?
Yes, getting back to the genetics, any indications of how long they've been isolated from the Alaskan population, and why they aren't intermingling with the Alaskan variety based on the migratory birds migrating annually?
Well, I'm fortunate to have present on this Zoom my colleague, Laura Corey, Dr. Corey here. She's a geneticist. She knows more about those things, but I don't know what's been done with the genetics on these different islands between these populations yet. I assume some things have been done, but I don't have an answer. But I would like to know.
Dr. Corey? Did you want to make a comment about that? Okay,
Yes.
you are muted. There you are.
I was having a little challenge unmuting. Good evening, everyone. So this brings back some fond memories. We passed the 10-year anniversary of March 11th, 2011, which was the triple disaster in Japan, which Larry experienced as the tsunami with his students. And one thing, to address one of the previous questions, one thing I remember from those reintroduction efforts, I did not get to go to Hawaii. I just sequenced things in the lab. But one thing I remember was how careful they were to set them in places that, and they had cages over them that protected them from feral pigs. So I would imagine that kind of technique would be used again. We spent some time trying to see isolate DNA and do sequencing and see what the genetic differences were between the different populations. And based on the kind of technology that we have accessible to us, which is single gene sequencing of chloroplast DNA for the plants, or for the ribosomal DNA for the fungi, did not see differences in that particular gene. So in order to really see differences between the population, you'd probably have to do something like next generation sequencing, which is a little too rich for our blood as a small college in the Midwest, but is also coming down all the time. So as the technology gets better, those experiments become much more accessible. Additionally, another tactic to take would be to look at similar different populations and see if there are particular genes that could be sequenced that would give that answer. And that is something I haven't thought about for a number of years, but I would like to do that again. And then I think I answered the question, but I actually had a question. Did I answer the question? Okay. One other thing is that Talizanella calispora, that's the excellent germinator, has been found all over the world, but it has never, that particular species of Schleizonella has never been isolated in Hawaii. But people tend to go and look for the orchids rather than going and looking for the fungus.
That's what I was going to say.
I come at this from an attitude, I'm really interested in the fungus. The orchids are lovely, but I'm really interested in the fungus. And I'm wondering if there could be efforts to, sample, specifically thinking about sampling for the fungus rather than sampling for the orchid, because the genetic sequence of the Calospora that's been found in Thailand is identical to what's been found in Florida, is identical to what's been found in other places in Asia. So it seems to be a fungal species with worldwide distribution. And if we knew that it was actually native to Hawaii, that might expand the opportunities for conservation germination.
Thank
Yeah,
you.
I guess the fear was that if you were introducing a new fungus, it could be an ecological disaster, but it may be here.
Absolutely, it might be here, it might not. And until we know that Calospora is, and I would even, you might want to do more than, you would want to know more than just, oh, it has the same, so to genetically identify the fungus, we use a particular sequence called the internally transcribed sequence. So it's just this little sequence that we use for identification, but then also morphological identification as well. So we'd want to know, really, is this found in this location? And then you could use it for reintroduction.
Well, also too, if we had that GPS of that place in Puna where the pig farmers were, then we could go and look for the fungus in that same area where those orchids had been. But yeah, I was thinking exactly the same thing. Why aren't we looking for the fungus? The fungus among us, right?
I have been. Yeah. Well,
And Fanish,
as Molly said, we tried a new technique of finding your naturalized orchids that aren't native to Hawaii and using them as biological filters to get us the fungus that's in Hawaii without cutting the roots up of the rare stuff there that's native there.
mm-hmm.
And we got 3. They looked good. They looked the part. Nothing.
I'm calling on. I'm going to call on Phoenicia now because she's been waiting and waiting and waiting. And so, okay, Phoenicia, you're up.
My husband was part of the group that did that orchid for the KOS and found it, and
The Kona Orchid Society.
and all of that. Yeah, I think he was president back then, but he was part of the people that went trudging. And it was actually up in Volcano Park, and they had to get a special permit to even be on that part of the land. It had to be approved. It had a special approval. And yes, they did find it and did all that. And I believe he said that at some point they went back, and I don't know if pigs had been in or what, but the area they couldn't re-find it. But he would know where it is. I mean, unless his memory is gone on that one. So, but he was part of it all back then. And he did the trudging and the hiking and the whole thing with Brenda and Larry and all that. So I could ask him. He's not home right now, but I'll ask him.
It'd be great to get that information, and perhaps in the next time the Illinois College crew is coming to Hawaii, we can schedule a couple of days to go and check it out.
I'm sure if Bob and Larry got together and talked about it, because they had a lot of laughs out of the hiking too. So I'm sure the memory is still there. So yeah.
Right, we can document another population of the Platanthera. Larry, I don't know if people are still wanting to ask you more questions about the Platanthera, but I believe you have some excellent news about another Hawaiian native orchid.
Yeah, I do. Thank you, Tom. I have 2 nuggets to offer tonight. Well, today, I guess where you're on, it's nightfall here. I believe it was Hank sent me an image that was forwarded from Lion Arboretum on Oahu. they used our protocol, the same one that Molly's using now. And they're cultivating now protocorms of Platanthera holochila. That looks so far so good. And also the other two. I understand that one of the Liparis that's there is now in cultivation. We have Liparis seedlings here. I have 2 students listening in right now that have protocorms of Liparis with and without fungi. The fungus is just to see if it works, and it's working. But Jack Lou's on the Zoom with us today, and he's a senior who's going to present his findings at the state of Illinois in another month on the propagation to seedling stage of the Liparis hawaiiensis.
Excellent.
So Lisa and Steve have been waving their hands. So what's the question? Hi.
Interested in the idea that you sealed your seeds in for quite a long period of time, way longer than we did at Fairchild. And I was wondering why and how you came up with that idea.
And why so long in the dark?
Yeah.
A lot of terrestrials, well, I wouldn't say a lot, not much is known, but some of the Platantheras on the mainland here are inhibited by light. And it's been thought that because it's a terrestrial, their seeds would be taken down about 4 inches down into the soil where it's dark. And it's a natural process. We have also worked with some Platantheras where if you blast them with 7 days of a photo period and then darkness, then they have a spike in germination. I think one of the problems that might have, one of the mistakes made early on, perhaps, with why this orchid in Hawaii is so difficult, was because maybe it was exposed to light, and as a terrestrial, maybe you shouldn't do that. And so we've always done the darkness first to get the carbon, enough carbon from the medium to get the leaf formed, and then you bring them out to mimic nature pretty much as close as possible. I'm just glad that we had seedlings that made it as far as they did as this terrestrial species, that they could be outplanted there in Hawaii. I never, quite frankly, I never used asymbiotic germination until this orchid told me to do that because it didn't want our fungi, except for one from Florida. So I'm just glad that we got them to grow as well as we did.
I'm a little worried about you, Larry, if orchids are talking to you.
I worry about myself all the time.
Mm-hmm.
Yeah, they talk to me all the time, Tom.
Well, non-verbally though. I mean, they're not actually like moving their lips, are they?
Telepathically, right?
There you go.
I don't know. I don't think so.
Do we have any more questions for either Molly or Dr. Zettler? Oh, yes, Jeff. Yep, you have to unmute yourself.
There we go.
There you are.
I was wondering what they look like. So just in case I'm out roaming around and I came across something that looked like an orchid, am I going to know that it's this one? I mean,
did you see it at the beginning, Jeff, where Molly showed a picture of what it looked like?
I saw the flowers, but the plant, you know, the chances are.
One of the first slides had very nice. She's going to share. There we go.
That picture Molly's showing you, with the genus Peristylus / Platanthera, you can find seedlings that are beginning to photosynthesize when the strap leaf emerges out of the sphagnum or the moss. It's going to be about an inch long. For the first time it begins to collect sunlight. It'll be, I don't know, it'll be about an inch long and maybe a centimeter wide. I shouldn't mix metric with imperial there, but about that big. And so, but you have to really know it. Sometimes seedlings of grasses can look similar, and but that's what you would look for the seedling stages, the early seedlings that are beginning to emerge. And as far as plants, the adult plants, you see the strap leaves shown there. They come out of the ground as one long strap leaf, and then when they flower, usually they put more up near the stalk. Sometimes two strap leaf.
Larry, what time of year would we normally see flowers of this species in Hawaii?
Flowers, I believe in the early fall is when we went. I'd have to check my notes, but we flew over to your islands, I believe in early fall. It's been 10 years, I should remember, but about the time when on the mainland, some of these terrestrials are being to set seeds too. It's not that much different.
Dr.
Oh.
Zettler, I believe one time it was spring break. I remember because I had students use your articles as references in a paper they wrote. And you brought the seedlings over in spring break.
Yeah, we brought seedlings there about a year, well, this time 10 years ago.
10 years ago,
But
right.
when we collect the orchid capsules, or when we usually get them in the fall, I think the capsules are ripening, and then it goes dormant. And I understand on Kauai, I've heard this from Steve Perlman, that there's kind of like a group of people that just wait and wait and wait every year for the first leaves to show up of that one plant that's left. There's a big celebration when it appears. And so I imagine it's going to happen soon there. I think that's pretty nice. It's really nice.
Do we have any other questions for our guests?
Lenora,
I
Dana.
Lenora.
I just have one question for Larry Zettler. What did one mushroom say to the other mushroom? Gee, you're a fun guy. I thought it would fit.
I've been the subject of ridicule many times about fungi.
No, that's not a ridicule. That's just you're a fun guy.
Cool. The Rotten Kingdom. I'll tell you guys a story. My wife may be listening. When we were dating, being a vegetarian, I had the infinite wisdom of we collected mushrooms, here in the Midwest, and we cooked them in my kitchen. And then I had the infinite wisdom of telling her, oh, did you know the DNA work suggests that the fungal kingdom is more closely related to animals than the plants? And she didn't like that. So I had to think quickly, which I did about, it's not a good thing to tell her. So I said, well, it's just the fruiting body. The fungus mushroom is what you harvest. The fungus itself is below the ground, decomposing things. And it's like the apple in the apple tree. You're not going to kill the fungus. So don't worry, everything worked out just well.
Oh, you're just
Well, I'm
eating.
glad I said the little joke. We got a good story out of it.
That's a pretty good one.
So Larry, now you have the combination of a fungal joke and a dad joke. Good for you. You can use that.
Yes.
Yep.
And I think I see. Okay, does anybody else have their hand up or waving? Oh, Lillian. Lillian does, yeah.
Lillian's not taking any chances. Oh, it looked like you had a mask on. But you
Oh, no, it's the light. Sorry. Actually, I did kind of have an interesting question to me, to both Molly and maybe Dr. Zettler. Is, as a gardener, not as a scientist, how hard is it to raise these babies, pass them on, and not get to see them go back to where you want them to, or follow through in that way?
Yeah, I mean, I do call them my babies, so I do feel I am a mother to them in some way, so I do have an emotional connection to them for sure. I haven't had to leave them yet, so I don't know how that'll be when I leave them. But I've had gardens before. I'm not very good as a gardener. So I've never had this much of an emotional connection and never grown something so rare. So I'm sure it'll be hard to leave them, but I hope it makes me a better gardener in the future.
they're going to be in good hands with additional students. But Molly, will you tell us what's next for you? Are you you're going on to graduate school? What are you going to do?
So when I sent you my bio before, I didn't know where I was going, but now I do know I'm going to Indiana University for grad school, and I'll be studying environmental sustainability there. So that's what's next for me.
Cool.
But
So what do you want to be when you grow up?
Sorry,
What are
hit
you
mute
planning
too
on
many
doing?
times. Yeah,
She's
so I'm
grown
looking
up, Dana.
to I'm hoping to work in the sustainability sector, so possibly as a sustainability director, just helping the world become more sustainable. Whether I don't know where that position would be, but in that facility.
Trying to get her to study orchids when she goes for a PhD someday.
That's right, Molly, because you're an orchid person now. Your old life is
Yep.
gone.
There's no
You're
going
an
back
orchid
now.
person. You've been inoculated, and therefore there's no going back to your old life.
I'm becoming an orchid.
Yes, you are.
And also, I don't know if you've heard this joke about the different degrees, but you know, bachelor's of science is BS, master's in science is more
BS.
more S,
Yes. Okay.
and PhD is piled high deeper. So now that'll be a mom joke that you can tell one of these days.
But keep in mind that when it's piled really high, there's lots of mycorrhizae in there.
Good point.
Oh, Tom.
When life
So
gives
any other-
you lemons, or in this case, BS,
Yes.
make fertilizer tea, right?
So do we have any other questions? Or, oh, yes, Jeff. And then I think, Matthias, did you have your hand up too?
Didn't Dr. White
Okay.
have another question?
Jeff
Can
has a
you lead us?
question. And then, Armand has a question. And then Matthias has a question.
I got a question. Okay.
Okay.
Of the other orchids, the Liparis, is that seen on the Big Island too? And there's two others endemic here. Are they on this island?
We've Liparis is on, is it Lanai is where they grow the pineapples? Is that correct?
Uh-huh.
Yeah.
Liparis is more widespread. Enectochilus is more widespread. I think Enectochilus is maybe the more common. Well, I don't know if they're really common, but more abundant of the three. And I'd have to ask Hank, he would know that, the specifics. And it's been a while since I've heard the latest on the distribution, but I think the Big Island has at least one or two, but not holochila.
Well, it might be here. We're going to go look for it next time you come.
Yeah, please do. I mean, if you guys hear any reports, or it would be really nice to let Hank know and others there, so that could be, staked off and some seed collected for propagation. That would be a really unique, wonderful find.
And for the members, Enectochilus is a jewel orchid that is apparently the most widespread of the Hawaiian species, but I don't know if it's on the Big Island or not. We'll check that out.
Okay. Armand. You had a question, Armand? Are you there? Okay, I'm going to move over to Matthias. Matthias, you had a question.
Can you hear me?
Uh-huh.
Yes.
Yeah.
Okay, good. I had to unmute. This might be a question that you probably don't want to hear, but as I'm listening and it's all about the seedlings, I mean, if it's so hard to do it with the seedlings, why don't you guys clone them? I mean, then you would get a really big population at once? Or is that something that you guys don't want to do because of genetic diversity?
Then, that, I think that would be correct. It's seed propagation is assuming that there are some genetic transfer going on. Cloning the few individuals that you have left, if that can be done through, I'm not one of those type of scientists, but we got to do something here soon. And I don't, having something cloned is better than not having anything at all.
Exactly.
So, these students that are on here, I keep telling them, number one, please don't let it die under your watch. Number two,
Pressure.
number two, I'm going to be at this maybe 15 years or 20 years more, and then they're going to have to carry this on. And it's going to be hard, a lot of challenges ahead, but I'm hopeful that there will be ways of growing orchids in a number of ways, and that we can get some of these to survive by the time they inherit the earth.
Larry, I was going to say, if this is a self-pollinating orchid, that sounds like the genetic diversity isn't very great to begin with.
Exactly.
So maybe cloning would be a strategy to get at least biomass reintroduced.
Yeah, there was some talk of actually having the Florida fungus grow some of these up for cultivation, say in a garden where the fungus isn't allowed to escape in Hawaii, so that you can get the seeds, and then you can keep going with that.
Mm-hmm.
So there's other options that we're cooking up that could possibly happen.
Great idea. Thanks, Matthias.
Yeah, that's a really good question. Armand, are you there? Yeah, he has his hand up, but I don't see him.
There's nobody there anymore.
Well, he's just been a blur through the whole meeting. Anyway, yeah.
Tell him to stop drinking so much. Just kidding.
Armand, if you're talking to us, you are muted.
Oh no, that's
okay.
So.
All right.
Unless there are more questions, maybe you guys want to start with a show and tell?
Yeah, we can move on to the show and tell portion. But maybe before we do that, we should give a big hand to Molly and Dr. Zettler.
Absolutely. Well done. And isn't it
Thank you
nice
so
to
much.
Isn't it nice to know what goes into protecting a rare and endangered plant like this? And
it's really interesting. You know what? Dennis came back and he has a question.
Oh, great.
Okay, Dennis. You have to. Oh, you don't have a question. Okay. You do have a question? You have okay, never mind. Okay. All right, so shall we begin our show and tell? Does anybody have orchids that they want to show?
I do.
And I think, the best way to see the orchids is to take your setting off of grid, if you had it on grid, and just put it on the person who's talking. So when I do that, then I can't see who wants to share. So who has an orchid to share? Venetia. Is the one behind you the one that you want to share? Oh, here comes Kathy. Okay, what you got there, Kathy?
Okay, I got this from Phoenicia the other day. She grows beautiful orchids. It's a Cattleya amethystiglossy. I pronounced it wrong. But
amethystiglossy.
Okay, I had to get something because I had made 2 trips to Hilo and I came home without any plants 2 times. That was a new record. So I had to get something. And it's really big and gorgeous. She grows beautiful plants. So
yes, she does.
When we're allowed to go back out onto the road, we have to do go visit Phoenicia's garden down in Ocean View and her greenhouses.
Yes.
Good.
Okay. Anybody else? Jeff? Oh, that's big. Do you have something that you want to show us for show and tell?
Yes.
Okay. Oh, what you got there?
Dendrobium Finisteria.
Finisterre.
Yeah, Finisterre. It's got a couple more ready to come out, but this one is the first one out yet. See how
Does it have
fuzzy they are? Yes.
Does it have a smell, Jeff?
Actually, there is a smell, but it's not unpleasant at all. It's just blah. I guess it's not sweet or anything. And,
Okay, somebody just said that they can't see any of the pictures of the orchids that people are sharing. So I'm going to say it once again. If you change your view from grid to just just one person at a time, then you'll get to see the whole orchid really nice and big. Okay.
There's a speaker, when you go to view, there's speaker view and gallery view. You want speaker view.
Right. Okay, so Jeff, you have another one there? Ooh.
Bulbo falcatum.
Whoa.
Gone.
Janice, did you have a question? Janice?
Jeff, if you speak, we'll still be able to see your plant.
Oh, here it is. Bulbo falcatum. There's a better view.
Wow.
They bloomed.
Boy, you sure do have a lot of flowers on that.
It does. It does.
Well, I'm sorry. Apparently there's somebody that still can't see. She said that she's on speaker view, but she can't see anything. So I don't know if it's just her own device.
So
It's because you are speaking.
Dan, another problem is every time you say something, then we see you. We don't see Jeff's plant. So Jeff has to talk and other people have to not talk.
Exactly.
Got it.
Like I just did.
Like you just did. Yeah.
Jeff, talk.
Jeff, and you're muted. And unless you talk, we can't see you in speaker view. You don't show up.
Okay, here I am. I unmuted and I'm back. But this is it. Can we see it?
Yes.
Okay. Above my name and okay. Yeah, it's pretty cool. This is the best it's ever looked.
Tell them to keep it outside.
I keep it outside in the rain. It's out where it likes to be. It's in one of those pods, Brazil nut pods, and been doing fine for 7-8 years since I had it.
How much light is it getting, Jeff?
It's under a citrus tree, so it doesn't get that much light, but I had it in another place before, and this is as good as it's ever been. So it's got quite a bit of shade, you know, it's a grapefruit tree. So it's kind of a lot of shade. Definitely not as much sun as the dendrobiums are getting.
How about that one behind you?
All right.
Maybe somebody else.
I also have this other one if nobody else is showing right now. And I think I had it last week also. This is something I got from Karen. This is something I got from Karen, and this is twinkles or something. Another dendrobium. Okay.
Okay, Dennis, you want to show us your orchid?
Sure thing. Thank you. This is, I think I got this at Akatsuka on your side of the island. And I live by the Kone Airport, which, as everyone knows, is one of the driest spots on the island. So it's hard for me to grow a lot of different orchid species, but Cattleyas do pretty well for me. And this is called Hawaiian Thrill Paradise. It bloomed for me just a couple of days ago. I like the Cattleyas. They're nice and big and they smell beautiful. So, and I always think of them as the orchids we used to give our dates when we went to prom years ago.
And I think, Matthias, is this one of yours?
Nope, it's Akatsuka
Nope.
breeding.
Oh, okay.
Actually, it's Carmela's breeding. But Akatsuka cloned it.
Do I hear a coqui frog in there?
Somebody,
Yes.
somebody.
That's real. Well, I hear they're a problem there, but last time I heard one of those was in Puerto Rico.
That's where they all came from.
That's right. Well,
I have one to show.
Oh, Lorena,
okay.
Lenora.
I know I
My
go
turn?
by, even though it says Lenora. So I have a, can you see me?
It's a little out of focus.
Am I on speaker
We
view?
can hear you.
Yep.
Okay. So this orchid I got at the auction at my very first holiday dinner with Orchid Society about 4 years ago. And it still blooms every year. It's beautiful. It has a nice smell to it, and it still has a Merry Christmas sign that it came.
Is that the secret of how to grow it? Is the Christmas time? Ah,
And I have it in my living room right now. And the name of it is Dan Microtrip and Roy Tokunaga. And it's just
Okay.
beautiful. It blooms every year with all these beautiful flowers.
very
Thank you,
long
Lori.
lasting.
Okay.
Thank you. Okay, Matthias.
Right. I hope everybody has seen the, finally seen the January issue of Orchids Magazine. I wrote a small article about a grower in Thailand, Mr. Noi Suvan, who is an incredible breeder over there. And I do buy some of his plants and grow them up from flask. And what he does, which I really admire about him, is that he breeds, of course, extensively, number one, and he grows out a lot of the seedlings. What's very hard with Cattleyas to achieve is to get a flower that is completely concolor, which means it has one color, lip, sepals, petals, all in the same color. So I finally have one, if I can figure out how to flip it.
To turn your phone around.
Yep, there it is. It's a Thaxina Gold, has an incredible shape. And as you can see, it's a solid color, lip, petals, sepals, all the same color. Actually, I have two of them flowering at this time. So this is really, really hard to achieve, to get a flower all in one beautiful color. And the other thing I really like about this one, as you can see in the lip, it's pretty flat. It has a little bit of a cleft lip right here, but it's almost 100% flat. So one of the parents is the Hardy Eye Delight, which is a big yellow with a ginormous lip. So you can definitely see this in this breeding. It's just an incredible flower. And just wanted to share that one with you. I have finally two plants flowering. So this flower did not open as good, but it is still pretty damn good. This one is a little bit younger, so it's more yellow. As they mature, the flower turns more orange as this one. This is about, I think it's now day number 8. So it's really an incredible plant. And even the foliage on them, as you can see, when they get a lot of sunlight, they have this little burgundy tinge to it, which comes out from the light and also, I think, from the cooler temperatures that we had, unfortunately, lately. Anyways, I wanted to share that one because of the article that was in the magazine as a follow-up.
Thank you. Do you have any others that you want to show us?
I have one seedling that is didn't come out as good. I'm not so happy with it. It's this guy here. I think you have one also. This is the Princess of Hawaii, my own cross. It's a big flower, but it's very floppy. But I do have another one that I'm really excited about, but it just opened today, so it's not fully there. I've been waiting for 3 years to finally see a flower. And so this is Toshiayoki by Bob Crowder. And it does exactly what I was hoping.
Oh my goodness.
So it's just opening. So Toshiayoki is the yellow one with the red lip and red stripes on it, the pizzazz. And the Bob Crowder is the orange one that Pauline got awarded. So I was hoping for an orange flower with red flares. And so Bob Crowder opens up brownish. And this is exactly how this one opens. It's brownish. And so I think in 2 days, it might be orange with the red flares because it does have the red streaks on it. So I'm really excited about this one. And there's another seedling here. This is a remake of Andon Treasure. So you can clearly see one of the parents, Golden Zell Lemon Chiffon. It's a very popular Cattleya in the background as a parent. It still has a little bit of that red streak that Golden Zell always has. But the shape is more rounder than Golden Zell, which comes from the other parent, Erin Kobayashi. And then this one, I don't know if I'm happy with it or not yet. This is one of my own crosses.
Oh,
so
that's
this was
beautiful.
Yeah, I'm not 100% there yet, I think. This is a large white. So this is Tiffin-Bells, a classic large white crossed with one of Ben's plants. His plant's called Golden Child, which is an orange. Well, this is what came out. So I don't know if the color is going to be different or what, but it's kind of like it's not pink, it's not white, it's kind of in between. It's a little bit of a rose color, I would say. It's not bad. It's just not what I was expecting, especially as the plant is kind of really tall, which comes from Golden Child, I think. This one just opening. Yeah, that's kind of all that I have in this greenhouse. So there's always something coming. I have a lot of my Princess of Hawaii. There's a lot of them coming. And I got a few blue vandas, just they're seedlings also, but really good color. As you can see, it's kind of all the same seed cross. It came out pretty even, beautiful colors. And there's one other one, just nice spotting on it. Yeah, this is a beautiful color. It's like a light purple or something. And that's it for today. Thank you for letting me share my plants.
Thanks, Matthias.
A little nod to Tom.
Matthias prepared this incredible show table for us a few months ago, and we completely ignored it. He's not forgiven me yet.
Well, I have a couple. Hang on. This one is Bulbophyllum Wariath White Fang. And as you can see, there are two flowers blooming on it. There were 3. And this is a real happy bulbo, and it usually blooms for me 2 or 3 times a year, sometimes more often than that. and the flowers are pretty good size. You know, I'd say probably about the size of a chocolate chip cookie.
Well, it depends
And
on who
then-
baked the cookie. There's some big ones, big cookies
And then
out
this one has both of these have rebloomed for me. This one is golden aya, and this has a wonderful smell. Wonderful, wonderful smell. It's more intense in the morning. I don't think it smells a whole lot. Oh, there's still a tiny little smell there.
Like
and
honey?
It's hard to tell what that smell is. Definitely smells like some sort of candy for sure.
Hyacinth.
And I love how it's, you know, it's pendulous and it just hangs down and kind of, you know, so pretty. And so those are just a couple of the ones I wanted to show you. So does anybody else have any orchids for show and tell?
I have an orchid.
Okay.
I don't see my,
You want to hold it up to your camera so we can see it a little bit better?
how's that? Is that better?
Nice.
Pretty good.
It's a Hocoglossum. I like it because it has the one thicker petal that is straight up and perpendicular to the other petals. Hopefully, I think I need a better background to really show it. And I recently bought one of its cousins and found that some people call these pine needle orchids because this is the leaf. See, these are the leaves and they're very, very thin. And I this one looks like it's maybe getting a little bit too much sun. I have it hanging right on the railing for my lanai because it likes sunlight, but I think it's, well, obviously it's too much because it's got a burgundy color, but there's the flower. That's this is
orchid.
About maybe 3 years old, and it's bloomed about probably at least 3 times. And it's been blooming for about 3 weeks.
Beautiful flower.
Well, thank you. Yeah, I just think I was surprised that I was able to grow it, but I took a chance. It's like a tiny little pot, and but yeah. And then I found out today that the tag is. I'm glad I knew where it was because the tag is all
Fading.
Yeah, all faded. Yes, thank you. Yes.
So Ellen, just a comment. So because it's blooming, it's probably getting the right amount of sun. It's not necessarily getting too much. I mean, the burgundy color on the leaves, that happens sometimes in some orchids when the leaves are getting a lot of light. But as long as it's not getting leaf burn, as long as they're not burning brown, then it's probably okay. And the proof is that it's blooming.
Thank you, Larry. I did not know that, and I was concerned. Thank you very much. Good information. Yeah, because these are more, these are kind of green. It's just, I guess, maybe the older ones. Thank you. You can see it there.
It's not a hard and fast rule, but often when plants have terete needle-like leaves like that, they are high light lovers. Often, not always, but
That's what I kind of thought when I and then maybe I read up on it when I bought it, but yeah, I was thinking that it had such thin leaves that it could take more light. So I put it where it would get the most light and still be protected from as much as I could. And I live in Mountain View, so that gives you an idea where it's a little, the sunlight is more subdued and it's not quite as hot.
well done.
I have one.
Ooh.
Oh boy.
Um,
You have to talk or we can't see you.
can you guys see me, Kathy?
Yes.
No? Am I on speaker view?
Yes.
Okay, so anyway, this is a Dendrobium linnea, and I got it at Pukalani Stables in Waimea from a guy that lives in Ahualoa, which is north of up the mountain from Honoka. And I just fell in love with it. Anyway, I can't even see myself, so I don't know if you guys can see me. So
Yes, we can see you.
move on to somebody else.
They're just doing show and tell.
Okay, thank you.
We ready to go?
So I have something here that Phoenicia grows in our landscape called Schlumbergia undulatum. The flower spikes are like 6 feet tall with these cluster heads. Kind of an interesting plant, just opened up this last week. And there's also this dendrobium that I'm not sure the name of it, but it likes it here in our elevation, 2,300 feet.
Wow.
That's it.
Great, thank you. All right, and anyone else?
This is from 3 weeks ago. Paplia maldii. It's growing on an Australian tune tree.
How did it look?
What's happening?
Yeah, it's growing on an Australian tune tree. And the trees, it's on the northern side, covered with lots of moss, lichen.
Thank you. Do we have any other, excuse me, any other people that have orchids to show? I did want to let you folks know that for next month's meeting, Matthias is planning on donating some honohonos to the people who are attending that meeting. Matthias, do you want to talk a little bit about that and explain what you would like people to do as far as keeping track of things? Do you want to talk about that, or do you want to wait for next month? Where did he go?
I had to unmute, I'm sorry. Well, we talked about that at the board meeting. So I'm giving away some of my hono clones. They're all lavenders, the regular lavender, very common here in Hawaii, lavender, hono. Okay. So what we wanted to achieve is that everybody who gets one to keep track of the orchid. So I will give out for everybody who wants to pick one up some care sheets so that they know what to really do. And we don't really want to do a competition type setting, but we would like to get every other month or every 3 months some updates on the plants so that we can see how they're doing. So I'm thinking of, the first update should be somewhere around August because the hono orchids need a lot of fertilizer and water during the growing season. And so we would like to get some kind of measurement so that we can see how good they were doing until then. And then by October, November, you need to stop with the watering. So we're going to do another update and make sure that everybody knows what to do at the time. And then, of course, we would like to see next year around April if you guys were able to get them to flower. It's just a little project, so to keep everybody, number one, entertained and engaged. And that's that.
Thank you, Matthias.
You're welcome.
So, yes. So something for y'all to look forward to for next month. Is Tom still here or did he go?
Who?
Tom? Miranda? Well, is he there? No? Well, I can't, if we're done with show and tell, and then I would just like to, we can adjourn. I think, let's give all of our Illinois people, let's just give them a big, a hooey ho or something, just something to let them know how much we appreciate them. If you want to go ahead and unmute yourself so they can hear you clapping, that's fine. But thank you so much for everything. And good luck to you, Molly. And good luck to your folks that are coming up next, and hopefully they don't kill your little babies. We'll have to get a progress report on that.
Mahalo, ahui ho, which means until we meet again.
Yes, until we
Aloha.
meet again. Ahui ho.
Aloha.
Aloha. Well, it's been a great meeting. Thank you all.
Thank you, Dana.
Thank you, Lori. Nice to see everybody. Okay.
Bye.
Bye-bye. Great participation. And thank you again to Matthias for his generosity.
Thank you, Matthias.
Mahalo, Matthias.
We love your orchids.
So if any of you Illinois people want to come to the meeting next month, you can get a hono hono.
Yeah, and you get to come to Hawaii to pick them up.
That's right. Okay. All righty. Well, I'm gonna hui ho myself here pretty soon.
Okay, I think we're gonna end the meeting. So bye everyone, and thank you for being here.
Bye, Larry. Yay.
Bye-bye.